Welcome to Nikolai's genetics lessons, and today we are going to talk about gene expression profiling. Why do we need this? As far as you know, many health problems are related to change in gene sequence, especially genes consist of introns and exons. Those changes that happens in exons, which leads to change in amino acid sequence of the proteins, and this may cause such proteins enzymes to be misshaped and loss of function or alter of their function, which cause certain genetic disorders. But also genetic disorders and our health can be related just to different expression of the genes. And let's take, for example, a case study. Shan, age 17, is more than 40 pounds overweight relative to his height. His parents and grandparents are all overweight as well. Allen is similar to Shan with respect to age, height, diet, and exercise habits, but he is not overweight. Furthermore, no one in Allen's family is overweight. Both Shannon's and Allen's families volunteered to participate in university study to identify genes that play a role in obesity. How will researchers approach this question? At the first step, we take samples from obese family and non-obese. Which tissues we would be interested? It's not brain tissue, it's not skin. It's going to be fat tissue and liver tissue, which plays major role in obesity. In the second step, we identify gene expression profiles in non-obese participants and in obese participants. In next stage, we identify genes that express differently in both these groups. For example, in obese participants, we may see that all these genes are suppressed and downregulated, and non-obese, they would show normal amount of production of messenger RNA and proteins. Or it can be more complex than this. For example, in obese group, gene 43 can be upregulated, next gene can be downregulated, then upregulated, downregulated. But in non-obese, we may see different pattern. But it should be recognizable. If we will see certain pattern, we can tell and predict that this pattern came from non-obese group or from obese group. And as long as we can recognize such patterns, we can develop diagnostic tests. And we also can identify new drug targets, and this is going to be drug targets, these genes. If we would be able, with the help of drugs, to change expression of these genes, we also would be able to affect how fat's metabolized, and we would be able to, for example, by changing expression of these genes, change metabolism of the people. So here's another slide where we can see what's going on the cellular level. So here's a cell, here's nucleus, and this is DNA within nucleus with genes on it. Of course, it's shown here that all these genes are on the same chromosome, but these genes can be on any of 23 pairs of chromosomes. But for simplicity, it's shown like these genes are on the same chromosome. And as you see, those genes which are on, they would produce messenger RNAs, which will be used by ribosomes to make proteins. And proteins in their own turn carry out major functions of the cell and its metabolism. And when gene is on and its protein or RNA product is being made, scientists say that the gene is being expressed. The on and off states of all the cells' genes is known as gene expression profile. We have about 10 trillion cells, and in all cells we have the same chromosomes, 23 pairs of chromosomes. And what makes all tissues different? Different expression of those genes. Some genes turn it on in some tissues and off in other tissues, so making each tissue unique. So just by looking at the tissue gene expression profile, which genes are on and off, we can tell from which tissue this genetic profile came from. But nevertheless, tissue analysis will catch abnormal expression patterns in those people who suffer from certain disorders, like obesity. So knowledge of such genetic profiles may help us making diagnostic tests and also targeted drug design. Protein blockers can be designed to inhibit obesity-causing proteins, and gene switchers that interact with DNA to turn harmful genes off or protective genes will turn on can be designed. And this is all for today. Thank you for your attention. Subscribe and see you in the next video. Goodbye.